Jay, and I'm going to be providing our sixth session of this webinar series. This is going to be on activated sludge. My name is AJ Barney. I'm a research engineer with the Southwest Environmental Finance Center. My expertise is typically in wastewater. I was an operator, I was a compliance officer, and now I work in kind of the engineering water. Before I jump straight into activated sludge, though, I do want to talk a little bit about biological wastewater treatment. So, biological wastewater treatment started in about 1890 when people started using microorganisms to treat that were already in wastewater to treat the wastewater. And what they started doing is using that technology to manipulate those microbes and utilizing it so we can just remove the waste and biological oxygen demand from wastewater, and then we can create cleaner water. Now, this first started with a trickling filter, and the first installation in the US was in the early 1900s, but those became secondary as the dominant biological treatment process those became the dominant treatment process in the 1950s until they were replaced by activated sludge, which due to advancements in like aeration and oxygen delivery became the new standard for biological treatment in wastewater. Now, with all biological treatments, like most biological treatments, activated sludge mimics mechanisms first observed in the environment, such as rivers and other bodies of water. Zoogleo is a film. It's a massive microorganism such as bacteria, fungi, and protozoa that grow on rocks and other surfaces in contact with bodies of water. So as water moves over the surface, there's oxygen that there's dissolved oxygen and there's turbulence in the water. And using that oxygen, those organisms feed on the organic matter as that water comes into contact with the film. Now, these microorganisms exist freely in the water as well and can grow in biomasses, and that's where our concept of activated sludge comes in. This is just real quick what that looks like. And you can see the water kind of rushing over, and as it rushes over those rocks, the wastewater, any pollutants or nutrients in that wastewater are going to be consumed by that zoogleo film or just the microorganisms that are already in the wastewater. And this is what those kind of original trickling filters look like. As you can see, they take up large amounts of space. They'd be in fields, and there'd be dispersal mechanisms to disperse the wastewater over those fields. But with the introduction of activated sludge, as you can see, there's a much smaller footprint. So this is why activated sludge, and especially with those technological advances, has overtaken those fixed film medias in terms of dominance of the biological treatment. And then before we jump straight into describing the activated sludge process specifically, just a couple quick biological terms that we should be comfortable with. The first is aerobic bacteria. And these are bacteria that live and reproduce only in an environment containing oxygen. They usually require oxygen to kind of break down nutrients and that sort of thing, and to consume food. Then there's also facultative bacteria. And these are bacteria that use dissolved oxygen or oxygen from other sources when the dissolved oxygen is not available. And that would be such as nitrates or sulfates. Now, these bacteria in our wastewater treatment process prefer that dissolved oxygen, but they'll use those other sources when it's not available. And they can live in aerobic and anaerobic conditions. Now, anaerobic refers to conditions that are absent of free oxygen. And then finally, the last one we're going to talk about is dissolved oxygen, which is just simply the molecular oxygen dissolved in water or wastewater. And there's a variety of ways to introduce that oxygen. So with that, we can actually talk about our activated sludge process. And this is giving you an overview of that process and an overview of how we use microorganisms to convert dissolved and suspended organics into settable solids. To start that off, why do we use activated sludge? Now, just fundamentally, it's to protect our receiving waters. If your wastewater system discharges into a body of water, we want to remove nutrients and pollutants that will be harmful to the plant and wildlife in the environment and any people that may live downstream as well. Now, we protect the receiving waters by removing our oxygen depleting organics and excessive nutrients. This is what we refer to as biological oxygen demand. Excessive organics in our receiving water increase the activity of oxygen depleting microorganisms, and this depletes the available oxygen for fish and other aquatic animals and then they can begin to die. This is what's called hypoxia, and it can lead to septic or swamp-like conditions in the environment where anaerobic conditions begin to take over. Now, excessive nutrients must also be removed, such as nitrogen and phosphorus, because excessive nutrients leads to things like algal blooms, which can produce harmful toxins that are harmful to wildlife and humans, and will also lead to more oxygen depletion in the environment as the algal blooms die off. This is an example of fish kill from excessive nutrients and a lack of oxygen in Florida. Then also with our activated sludge process, it's also important to remove solids. The solids contain the majority of our biological oxygen demand, the nutrients, and they also act as places that pathogens can hide. It's important to reduce pathogens, which are small microorganisms that can cause disease because they lead to waterborne diseases in human beings who may drink the water, play in the water, consume the water in other ways. Common disease-causing pathogens are things like bacteria, viruses, protozoa, and can lead to parasites like Giardia, Cryptosporidia, typhoid fever, cholera, and hepatitis. Those are our most common waterborne diseases. So that answers the why, and then we'll go ahead and talk about the what now, which is our overview. And as we begin that conversation, let's explain kind of where the activated sludge process fits into the larger scale of our wastewater treatment process. So basically, we start with the plant influent that enters the wastewater treatment plant. And the first thing we do is we introduce some pre-treatment. This is typically where we screen for trash, for grit, and we remove those items that can be harmful to the receiving waters, but also harmful to the equipment in the wastewater plant. This is followed by primary treatment. This is where we remove a majority of our solids from the wastewater. These are the solids that are too heavy to keep floating. So we remove out our solids in the form of primary sludge. And after our primary treatment is where our biological processes, and in the case that we care about today, our activated sludge process. And the first component of that is the aeration tank. This is where a majority of the biological activity is going to occur, and we are going to be removing our organics that contribute to our biological oxygen demand. So here we kind of show the oxygen being introduced into the aeration tank. It can't be an aeration tank without air. And this is typically followed by our secondary clarifier, which is used to remove solids that are created and were still in the aeration tank. And this along with the solids from our primary clarifier, both go to our solids handling facility and are removed from the process completely and either landfilled or composted. Then, along with the secondary clarifier, there's also the return activated sludge, which are the solids that are returned to the activated sludge process and reintroduced into the aeration tank. The other component of those solids is the waste activated sludge, which is just disposed of with our other primary sludge. So this leads to biosolids from, possibly like a digester or land application. And then sometimes, because in solids handling, we also do dewatering, there's also the internal effluent recycling, which is water from those solids that are returned to the plant influent and are reintroduced and retreated with the rest of our wastewater. Next, after our secondary clarifiers is just our water, our treated water from the biological process that is disinfected. And then all of that leads to the effluent, which is sent to our receiving waters. Now, the activated sludge process consists of this area right here, the aeration tank, the secondary clarifier, and then the sludge that is disposed of from the secondary clarifier and is either recirculated into the activated sludge or is wasted. So just a little more specifically, we're going to discuss some of these components a little more in depth. First, there is the aeration tank itself. This is where our biomass is stored and is cultivated to remove our BOD and other nutrients. It's called an aeration tank because this is where we provide the oxygen that microbes use to break down the organics in the wastewater, and the dissolved oxygen always, can provide mixing for the tank as well. And then also in these aeration tanks, there's typically mechanical mixing as well. Next, there's the biomass, which is made up of all kinds of very small organisms, such as our bacteria, fungi, archaeans, and other small animals, such as protozoa and amoeba. This, along with our solids and the components that make up our biological oxygen demand, make up the sludge component of our activated sludge. And the biomass, the small microbes in the system, are actually the ones that do the heavy lifting of the activated sludge process and break down the nutrients as long as we provide them the right conditions and a food source. So this is just an equation that kind of shows the chemical representation of the bacterial digestion process for treatment. So like we've been discussing, activated sludge primarily works through biological oxidation of the organic matter in an aerobic environment, which this equation shows. So we have the organic matter, which is the wastewater, plus oxygen, the other nutrients that are in the wastewater, and then our microbes. And when we add those together, they are able to cellularly respirate, and this creates, they reproduce and create new microbes. The byproduct of that cellular respiration is carbon dioxide, and they're also producing water. So in other words, the microorganisms' metabolisms that are treating the effluent, and we are primarily trying to keep track of the rates of the food entering the system, the solids leaving the system, and the bacterial growth. So the aeration tank is designed and operated in a manner to promote the growth of that bacterial mass in a way that it can flocculate and clump together and then can be settled later out of the clarifier. So with that, there are basically three perspectives that operators should be familiar with when thinking about aerobic oxidation. So the first one that we talked about was kind of the bacterial standpoint, which is the food plus the bacteria plus the oxygen. That's how they reproduce, and that's one that we need to understand. And then there's also our operator standpoint, which is we have our wastewater and the waste in the water. We introduce it to our activated sludge, we aerate it, and that removes that waste. And then also the last part the operator should understand is the chemical reactions that are going on. We have an electron donor, which is going to be our oxygen. We have energy, which is going to be the food and biological oxygen demand that the bacteria are using. They are reproducing through that process by consuming those organics and growing big enough and to reproduce. And then there is the electron acceptor, which is going to be the oxygen, the dissolved oxygen that we introduce into the tank. Sorry, when I say electron donor at the beginning of that one, that's actually the food that's part of the energy, and it's something that we take off the food as part of our respiratory process as biological beings. And basically what this means is our end products are our oxidized wastewater, are more bacteria which reproduce through growing and consuming food, and then also byproducts of carbon dioxide and water. So with that, there are a couple of different bacteria, which we kind of introduced a little while earlier, that are kind of performing these processes. The first is our aerobic bacteria, which, as the name suggests, they need to live in aerobic conditions and they need oxygen. They are known to grow relatively fast. They do not produce a whole lot of odors, and they are efficient waste oxidizers. So they break down that food and create those, they produce those byproducts like carbon dioxide. Now, aerobic organisms, as you may kind of assume from what we've been talking about a bunch, is they are the most prominent organisms in an activated sludge plant. The aerobic bacteria need that molecular oxygen, and this is why we pump air into the activated sludge tank to make sure that they have enough dissolved air to perform their function. Now, typically, these organisms do need a proper dissolved oxygen level to function. This is typically considered 1 milligram per liter for activated sludge. And without that, this wouldn't be an activated sludge could not perform the way it's intended. Next, we have our facultative bacteria. Facultative bacteria can use oxygen from the air, but can also strip oxygen out of compounds, like we said earlier, such as nitrates and sulfates. They do, like we said, prefer basic aerobic conditions because in those conditions, they can generate more energy than in the anaerobic conditions. But they manage just fine without the oxygen. Now, these are typically useful when our process is modified to remove things like nitrogen from the ammonia. That is also typically associated with wastewater. This is a picture of an E. coli, which really isn't used for too much of our processes, but it is an example of a facultative bacteria. And facultative bacteria, along with aerobic bacteria, work together to treat the wastewater in our system. Then another type of bacteria in our system is what we call filamentous organisms. And these are bacteria that can be detrimental to the activated sludge process or can help it form more effective flux. There are about 30 different kinds of filamentous organisms commonly found in wastewater. Some examples of filamentous organisms include Nocardia, Theothrix, and Actinomycetes. And like I said, some of these filamentous bacteria actually provide the support structure to help bacteria flock together. But if these bacteria are allowed to dominate the biomass will not settle correctly in our secondary clarifiers, which can result in poor effluent quality and floating solids. This is why it's important to observe our wastewater characteristics and understand what proper flocking and foaming should look like, so that these kinds of organisms can be identified and issues can be addressed immediately. They can also often be minimized using correct process controls and can be addressed with things like chemical treatments such as bleach, polymers, and other flocculants. Some recent technological advances actually introduce a reactor ahead of our aeration tank that actually prevents and creates an environment that suppresses the growth of filamentous organisms before they should be grown. Then along with wastewater, like most of our wastewater system, there are a variety of other types of organisms that exist. These are often examples that we can see in microscopes. These only make up about 5% of the biomass, so it's not always critical to know them, but it is a good experience when we use our microscope analysis, which is an important part of our process control. We can see some of these examples, and it's often cool to see things like a water bear and stuff like that. But yeah, these do make up less than 5% of the biomass, but there are these other organisms that exist in there. And they may be, predators of other things, smaller microorganisms, and they can indicate a couple different things. They can be used as an early warning sign that can help prevent critical upsets, because they indicate the age of your sludge. There's typically organisms that only exist in older sludge, and there's typically organisms that only exist in younger sludge. So these are some of those examples of those. It's important to understand those as well. Some examples are things like rotifors, which only exist in older sludge. So after understanding our biomass and the aeration and our aeration tank, the other component, or one of the other components that we mentioned in that diagram, is the secondary clarification. Because after biological treatment, the solids need to be removed. So this is where that secondary clarification comes in. These work the same as our primary clarifiers and allow sedimentation to occur to separate the treated wastewater from our solids. But a big difference between our secondary clarifiers and our primaries is that the effluent from the secondaries is much cleaner at this point. And as you can see from the picture, some people even have things like fish and other wildlife that may live in a secondary clarifier. I wouldn't recommend humans interact too much with this kind of wastewater because it still can have pathogens and stuff like that. Now, the secondary clarifiers serve an important role in the management of the solids in the aeration tank. Like I mentioned earlier, we have our return activated sludge and our waste activated sludge, and properly controlling the balance of those two is essential for controlling the process of the activated sludge and how that's going to perform. And we'll go into a little more detail on that a little bit later in the presentation about how we do that and what characteristics and like math is important for that. Then the final component, kind of broadly, is the sludge waste and return. So this is our solids handling. So when the sludge that is not returned as return activated sludge, we call it, like we said, the waste activated sludge, and that must be disposed of correctly. So we might have things like our solids handling. This is an example of a sludge belt thickener, which plays an important part in sludge waste. But these are just important to make sure that we can remove excess water from our sludge, and the sludge can be disposed of correctly in a landfill or composting and a digester, stuff like that. So that concludes our overview, but that brings us to our first poll question. And Matt, if you can go and bring that up, and then I'll go ahead and read it once it's up. I can't see it from, oh, there it goes. So our first poll question is, microorganisms use oxygen to remove what from the wastewater? And the options are DO, chlorine, BOD, and grit. And I'll give you guys about 30 seconds to answer that. Good. It looks like it looks like most of us answered, and it looks like most of us got the correct answer. The correct answer is BOD, which stands for biological oxygen demand. Because if you guys remember, those microorganisms use oxygen to remove that biological oxygen demand. If it was a little confusing with DO, DO is actually what we introduce as the oxygen for those microorganisms to use. Now, chlorine can be removed from wastewater with oxygen, but it isn't microorganisms who are performing that function. And then it's not grit either, because that typically takes a physical process to remove from wastewater. Good job, everybody. So with that, let's go ahead and talk more specifically about our activated sludge operation. Matt, are you able to pull down the question, or is it just showing up for everybody?
That's just you, AJ.
Oh, okay. Sorry about that. I thought it was still up. Okay, so our activated sludge operation. So this is just gonna be a kind of step-by-step piece of what is actually going on. Because we know biologically what's going on. You know, sludge is being introduced and we use oxygen to remove it. And now we'll actually talk about like operationally what's going on. So the first step is the introduction of our sludge components. So this is made up of our influent, which is going to be our wastewater with solids and our biological oxygen demand, our nutrients and stuff like that. And then also the RAS, our return activated sludge, which is going to be coming from the secondary clarifiers. So these are pumped around the plant. Typically, there's a lot more influent than RAS. But they are combined together into like kind of the front of a plant. This kind of shows isn't a great picture, but this shows the front of an aeration basin with some pipes going into it, and those are going to be my two types of influent, my influent and my RAS. And at the beginning step, they're going to have some initial mixing when they come into contact with like a mixing box. As you can see, that's our kind of some influent pipes right there. So these are typically big open chambers where the influent and the RAS are mixed, and then at that point it becomes our mixed liquor. We may have some mechanical mixing. This is the example of a mixer, and this is like a big fan underneath the water. Along our aeration basins, we typically have things like control valves, which are going to control the oxygen and the mixed liquor coming into our activated sludge. Then there's our aeration zone, where we have our mixed liquor mixed by our oxygen, and then our biological activity is taking place. Now, this oxygen is introduced by our diffused air. So these are typically created by blowers, and they're introduced through things like diffusers or aeration stones. And these are units and systems that are specifically designed to release a continuous supply of bubbles into the wastewater. That air transfer allows those microorganisms to use the oxygen to break down the organics in the wastewater. And with that, I have a short little video that I'm going to show that is going to kind of show that process and the blowers where they come from. This is an animation that one of our colleagues, Clayton, made.
Oxygen is a vital component in supporting all kinds of life on our planet, even tiny microscopic life. These microorganisms use oxygen to multiply and can be utilized to help clean water as part of the wastewater treatment process. But how does that work? At the wastewater treatment plant, there are large basins filled with wastewater. Oxygen is introduced into these basins to support the bacteria that will remove harmful compounds from the water. This is known as aeration. Aeration blowers create a vacuum, pulling air from the surrounding environment into the blower, where it is then discharged through outlet pipes. As air is discharged, the pressure increases, forcing the oxygen into the basin. Aerobic bacteria in the water use oxygen as a fuel to multiply and consume harmful contaminants such as organic matter, ammonia, and dissolved metals. The microorganisms convert these contaminants into less toxic byproducts, such as carbon dioxide, nitrates, and metal precipitates. Utilizing aeration in the wastewater treatment process breaks down organic compounds, reduces odors, and removes harmful contaminants. Crucial steps to cleaning wastewater before it is reintroduced back into the water system.
For more water and waste
So as you can see, this oxygen, like we said, is produced from a blower, and it's typically just oxygen or that's in the air, that's in the environment around it, and then it's introduced into our basins, and then bubbles up nicely like here. So that's what it looked like in the animation, and hopefully that was a good representation to kind of help you guys visualize what's happening here. But after kind of treatment, you have your wastewater that should be mostly void of your biological oxygen demand, or a large part of it has been removed along with other nutrients. And then what it does is it passes over kind of a weir system and will combine into a common chamber where all that wastewater can then pass on to our next step, which is our, like we said, our secondary clarifiers. So the secondary clarification allows the rest of that brown stuff in that mixed liquor to settle out and basically provides us with our clear water that can go over the weirs. So like I said, the big difference between the primary and the secondary clarifiers is this nice clear effluent. And you can see at this point, the clear water passing over the weirs, it looks like the same water that you'd see in a river. There's probably just more pathogen, and it still needs one more step in the form of disinfection to finish being treated. Now, the other component of our secondary clarifiers is the secondary clarifier sludge, which I've referenced a couple of times as return activated sludge, or RAS. And this sludge is the sludge that is pumped back to the activated sludge process. And then we also have our waste activated sludge, which is the sludge that is removed from the secondary clarifiers and activated sludge process in general and pumped to solids management process. Both of these control the blanket of sludge in the secondary clarifier. You want to make sure that blanket in the secondary clarifier is not too high, or it adds back solids to our treated effluent that can act as, like we said, hiding places for pathogens, or just exceed a regulatory permit requirement. So with that, we have our second poll question. Matt, if you could kindly bring that up. So this question reads, dissolved air is produced by what for aeration in the activated sludge process? All right, it looks like the majority of our participants have answered. All right, so if you can see here, it looks like most of us got it right. The dissolved air is produced by blowers, which our nice little video showed us. UV lamps are not associated with dissolved air and are typically used for disinfection. The anaerobic bacteria do not produce the dissolved air, but instead utilize that dissolved oxygen. And then scraper arms are used in our primary and secondary clarifiers to maintain sludge blankets and ensure sludge is funneled to solids handling correctly. So good job, everybody. And that takes us to some of our next section, which is an explanation of the different variations of activated sludge. So there are a lot of different variations or modifications to the activated sludge process that can be made, and a lot of different ways to classify them. So this can be based on our loading rates or the fluid pattern, such as complete mixing or plug flow, the reactor arrangement itself, the method for aeration, or the method for which solids are separated from the activated sludge process. And then finally, whether you are going for biological nutrient removal, or if you're going to include nitrogen and ammonia removal as well. So I'm just going to touch on about 3 of these. the most common ways to kind of divide those up. So the first variation is our conventional plant, which, like I'm showing you here, just kind of isolates the aeration tank, the secondary clarifiers, and the sludge that's produced in the secondary clarifiers. So this is a layout that I showed you in the beginning. This is a layout for that conventional activated sludge process. Now, a conventional plant is operated so that the amount of food available is kind of limited, requiring the organisms to compete for the available food, and is kind of characterized by the following operational standards. The loading rate is considered a medium loading rate. The typical sludge age is 5 to 15 days. It can handle a loading of 20 to 40 pounds of BOD per 1,000 cubic feet. And then it typically operates at an F to M ratio of 0.2 to 0.5. Some other components that are important for this is there's often a hydraulic retention time of 6 to 8 hours. So that will be our detention time for the air activated sludge basin. The organism growth in these are considered slow, and you kind of had a middle range of the sludge age and the organisms that are going to be in there. And this typically provides a very good effluent rate that would only need some disinfection to be dispersed or to be discharged into the receiving water body of water. The next is our high rate activated sludge process. And the difference is is that our activated sludge is going to be the first step in the secondary treatment and might, for example, be paired with another activated sludge process that is modified with an anoxic component for nitrogen removal. So some resources give the sludge range for this as being typically 1 to 3 days old. It can typically handle more BOD than our conventional activated sludge plants with 100 to 1,000 pounds of BOD per 1,000 cubic feet. Typically operates at a lower food to microbe ratio as well, 0.5 to 1.5. No, I'm sorry, it operates at a higher food to microbe ratio. So it's considered a high food to F to M ratio. Also, as you can tell, the hydraulic retention time of these are lower than the conventional activated sludge at 1 to 3 hours. And the goal is to grow microorganisms very quickly and get them out of the system. And then the effluent quality is not often very high compared to our activated sludge because more solids, there's less time spent, so there can be more slugs that are able to pass on to our other processes. And the final variation that we're going to talk about is the extended aeration activated sludge process. Typically the waste activated, or it's organized the same way that our other activated sludge would be arranged. And these are typically suitable for smaller communities that can kind of have a lot of space and can have a large chamber or a large channel that can facilitate this type of aeration. These can also typically handle these are great for smaller communities because they are able to handle flow variations and can handle like a shock load. So even though they handle like a smaller amount of wastewater, because they are extended and because you typically have flow equalization and longer channels for wastewater to be treated, if there is an issue, it can usually be diluted out and that can be extended. Also, sometimes with these types of plants, you may eliminate the need for a primary clarifier, and then you can just use a secondary. So a lot of times, our extended aerations are kind of similar to our package plants. Now, some of the this shows some of those same characteristics then the other characteristics is that as you can see here, the sludge age is 20 to 30 days. So that's much longer than our high rate or our conventional activated sludge. The BOD loading is similar to our traditional activated sludge, but is a little lower at 10 to 25 pounds of BOD per 1,000 cubic feet. And then it operates at a lower F to M ratio of 0.5 to 0.15. And it has a much higher hydraulic retention time of 18 to 24 hours. And typically, the growth of our organisms is very slow, but you do produce good effluent quality compared to our high rate. So this is just a quick table showing them compared to each other. Like I said, you can see a couple examples that, the low rate activated sludge process has a much higher hydraulic retention time and sludge age than the others. But then like the high rate activated sludge process can handle more of a load of wastewater. Now, then the other way to kind of describe the difference between some of our activated sludge processes is complete mix versus plug flow. So complete mix does just exactly what that name implies. There's typically mechanical mixers that fully mix the fluid in the tank and result in a very standard water quality throughout that is being dispersed. These types of tanks are typically round or square, and the aeration may be mechanical or with diffusers. Now, our plug flow system, this is kind of where similar to, or our plug flow would look like our extended aeration type systems. There isn't a whole lot of lateral mixing, and the fluid moves through the system as a plug. And the systems are usually rectangular or kind of work in a serpentine pattern that is a long track to create a smaller footprint. Now, for our plug flow, the BOD and chemical oxygen demand will be higher at the front of the basin and decreases as it moves toward the end, while biomass will increase from the beginning of the tank to the end. Now, our high rate aeration can be done with completely mixed or plug flows, whereas our conventional activated sludge processes are typically plug flow, and our extended aeration, like we said, is typically going to be that plug flow process as well. So this is what that plug process may look like. You can see that there's kind of that serpentine going on, and the water is moving from the front and has a long time where it's in the activated sludge process. And by the time the effluent is going to look much different at the beginning of this type of activated sludge process versus the end. So it starts at the beginning and kind of moves around that corner till it gets to the end process. Now, a completely mixed process looks more like this. Those round tanks where it comes in, it's completely mixed, and the water at the front or the beginning of the process versus the water at the effluent of the process is going to be very standard with the water at the end. Then there are some combinations of these types of devices or types of processes, and this includes our oxidation ditch. The oxidation ditch is a combination of the two, where part of the track is set up as a completely mixed. So when it reaches the end and it's turning the corner, that mechanical mixer will mix up the wastewater to increase the standardization of the wastewater amongst the whole wastewater flow. That kind of shows that wastewater mixing in there when it reaches the end of that serpentine. Then a final modification that we typically see is nitrogen removal for wastewater. This is typically a two-step process where we have our nitrification, the aerobic process, that converts our ammonia into nitrite and subsequently nitrate. Now, in order to complete the process of removing nitrogen from wastewater, you need a denitrification process, which is typically anoxic and converts those nitrates into nitrogen gas. Now, we don't go too much detail into it in this presentation, but you typically need to follow, if you remember our activated sludge process, we have oxygen, and then it's followed by clarification. So where do you actually have a time for an anoxic process? This is typically done by either adding an additional activated sludge process or using things like a Lutzak editor process, where you recirculate a lot of that treated wastewater that has nitrates in it to the front of the aeration basin, and you have an anoxic zone there where those facultative bacteria will not have access to the oxygen anymore and will utilize the oxygen and nitrates, remove it from the nitrates, and convert it to nitrogen gas to complete its BOD removal. Now, conventional activated sludge processes that run with a longer aeration time often result in nitrification, but the denitrification process has to be, like we said, separated. Another alternative form of wastewater processes that's kind of gaining some traction and gaining in popularity is the integrated fixed film activated sludge process. So these are what's called moving bed biofilm reactors. This will typically combine those fixed film processes with an activated sludge process. What you might have is like a biofilm carrier that is placed in the aeration basin and allows oxygen to go through the activated sludge, as well as microorganisms that on that biofilm. The benefits of these kind of systems is that there's an increased efficiency and a decreased environmental footprint. However, there are increased maintenance associated with the media, and those balls can kind of, or like the carrier, which is typically in the form of like a ball, can break down, and there can be bioplastics and more trash in the wastewater. Now, with all those forms, I'm just showing again, I want to show again the kind of our design characteristics table. So looking at our loading ranges, the SRT is right in the typical range for a conventional plant when we have about 3 to 15 days of that SRT, whereas our other processes are a little bit longer. I'm not going to make you guys memorize this, but it's important to remember some of this stuff for when we talk about some of our math later. So that leads us to our next poll question. Matt, if you wouldn't mind loading that up. So for this question, we're asking, ammonia is removed from the wastewater in activated sludge through what biological process? Good. So it looks like we've kind of peaked on the number of answers we're getting. So if you guys want to check this out, it looks like most of us got it correct again, 74% with nitrification. So nitrification is the process by which ammonia is removed from wastewater and converted into nitrates. Anaerobes are just are just microorganisms that can live in an anoxic environment, so without oxygen. Sedimentation is more of a physical process that removes solids and not ammonia, because ammonia is dissolved in our wastewater. And then if you remember, disinfection is how we remove pathogens from the wastewater. But good job, most of us got it right, so we're paying attention. Now that we kind of know about activated sludge and the different kinds of activated sludge, let's go ahead and talk about our process controls. So the reason these are important is because in order to operate activated sludge properly, we need to understand the characteristics of our process. And we've talked about some of these things like our foam, um, our RAS and our WAS. But we're going to go a little more in deep into that now. And because activated sludge processes are those biological processes like we discussed, they will have more variables than our typical physical processes that we see in wastewater like sedimentation or screening, and even more than our chemical processes like disinfection or coagulation. So operational data must be collected upstream and downstream of the activated sludge process to make process control adjustments in order to meet the activated sludge process objectives and also to account for any influent conditions that may upset the activated sludge process. Our characteristics of our influent and our activated sludge change daily, and the sludge process objectives must be maintained in a balanced state to account for those minor or major changes. Operators must also be ready for the unexpected and understand in detail how their specific process can be controlled to make emergency process adjustments. So the objective of our activated sludge is to maintain the aeration tank to reduce flock-forming bacteria, ensure that flocks are forming and that those flocks settle inside the secondary clarifier, and then also to manage the activated sludge to maintain a healthy proportion of microorganisms in the system, specifically in the aeration tank itself. We have a couple different types of characteristics that we want to look out for in our activated sludge. Like we said, we're collecting a bunch of data, but we're also making observations. So we have our observational data, such as things we're going to see, things we're going to smell, stuff like that. And then next is our laboratory characteristics, things that we can see in a microscope, things that we can measure with our standard methods. And then also our mathematical characteristics, things we can calculate using our observations or our laboratory data. Macro analysis of any microscopic analysis of any biological system should be a critical component of any ongoing daily monitoring and control. Lab sheets to correlate the health of the system, any changes of flock structures, higher life forms, filamentous identification, and suspended solids can be determined by using a microscope and examining the biomass. So because biomass is such an important component of our activated sludge, this is an essential tool that must be used to understand what is in our sludge. So this is typically done by taking a sample and putting it under a microscope. Then we do have some other activated sludge variables. Some examples of those variables that go into our influent or our activated sludge itself include dissolved oxygen, our F to M ratio, which is our food to microbe ratio, our mixed liquor suspended solids, our mixed liquor volatile suspended solids. I guess I'm missing a word there, so sorry about that. It should be MLVSS, which is the living portion of our suspended solids, our solids retention time, the mean cell residence time, or the sludge volume index as well. We will be going over how to calculate some of these later in our presentation. So on top of what's in our system, it's also important to understand the influent that will be treated by the activated sludge process. Analysis of influent characteristics helps with treatment and making our process control adjustments. Certain influence traits such as infiltration and inflow, high septic loading, and the different type of wastewater discharge from industries can have effects on the activated sludge process. For example, infiltration and inflow can result in excessive grit and silt that is washed out and may damage process equipment and can interfere with things like aeration. Higher flow in the collection system can also result in the washout of organic buildups in the collection system that can increase the treatment requirements of the activated sludge process. It may require things like more oxygen, or for us to manipulate our sludge that we're returning, or open up new basins, stuff like that. Infiltration and inflow can also cause temperature changes in the influent. This is usually colder rain that infiltrates the system and decreases the temperature of the water in the activated sludge process and inhibits normal operation. Then there's also the threat of septic shock from the influent system. When lines are blocked due to maintenance or due to damage, the wastewater can become septic, and when it is free again to flow through the collection system, it can also cause problems to treatment processes. Then finally, it is also important to understand what types of industries may contribute to the collection system and impact the activated sludge process. Those influent characteristics are going to be things like flow rate, total suspended solids, volatile suspended solids, biological oxygen demand, chemical oxygen demand, total kilodalton nitrogen, which is our TKN, ammonia, nitrates, temperature, pH, and alkalinity. Typically, characteristics monitored for those typically want to monitor these activated sludge, these influent characteristics, and we want to understand when they are changing. A lot of times these are can be done with monitors or through our lab analysis. Some typical lab analysis that is done to monitor those influent characteristics measure things like our BOD and our DO, pH, alkalinity, and toxic substances. SVI is also typically done in a lab environment, as well as our MLSS and MLVSS. And then our FTM ratio utilizes some of those other numbers to provide that FTM ratio. So those are the things that we can track in the lab. Now, typical ranges for those lab characteristics are listed here. A common one that so it shows when we get raw influent, what our COD, BOD, suspended solids, or dissolved oxygen might be. Then we have also, what it would be in our effluent and what it would be in our activated sludge processes as well. A typical one to be familiar with is like our BOD influent. It's typically a lot higher than our effluent, and we may see 150 to 400. That is typically decreased from primary clarification. And it's good to understand what's coming in because we use our BOD in things like our SRT calculations. Here's some typical, here's those ranges again, before we start going over, our mathematical characteristics. If you'll, think about these, specifically sludge age and FTM ratio as we kind of move forward into, after our observational characteristics. So these are examples of our operational characteristics. Healthy activated sludge has some easily observable characteristics that operators can use as indicators to identify when problems may be developing in the activated sludge process. As with most processes, and you'll hear me iterate this a lot, each plant will have their own unique characteristics. And it's important for operators to understand what their plant is supposed to look like when it's running well. But many plants share these common observations that we've kind of listed here, and they offer a good starting point for what to look for, or may even offer some insight into why wastewater treatment plant isn't operating correctly. So the first characteristic we'll talk about are the visual ones. And the first one that we like to discuss is color. So healthy activated sludge typically has a brown or gray color. The color may vary depending on factors like the composition of the influent wastewater, but it should not appear green, black, or exhibit other unusual colors. If it's black, it's probably septic, and it's going to be you're going to have a hard time introducing dissolved air to function the way you want it to. Next, there are the bubbles. Air bubbles from the diffused air system that provides dissolved oxygen to the activated sludge system should produce fine, crisp white bubbles. Then there's also we also have the visual structure of the flocks found in activated sludge. This can be viewed, like we said, with a microscope, but you can also get an idea of what your flocks look like by putting a sample in a clear, wide mouth beaker. So healthy activated sludge forms well-structured and dense flocks or clumps of microorganisms. These flocks settle easily in the clarifier and aid in the separation of solids from the treated wastewater and form a distinct and dense sludge blanket at the bottom. The supernatant, the liquid that is above those sludge, should be clear and free of suspended solids. So you can view that when you take a look at like your secondary clarifier. And if there's lots of solids, you may not be flocculating correctly in your activated sludge. Next, there is the olfactory observations, which is your sense of smell. So things you can smell. Healthy activated sludge often has an earthy odor that smells a lot like fresh or moist soil or dirt. This is a common odor associated with the metabolic process of microorganisms in the treatment system. And then also in well-functioning activated sludge processes, foul or offensive odors such as sulfurous or putrid smells are typically absent. So if you do detect the presence of those odors, it may indicate process upsets or issues. So I think that's so this is what that whole process kind of looks like. You know, those bubbles are real clear, the flow to the top right away. But this is an example of a good oxygen system. So then once we move past our laboratory calculations, or I'm sorry, our lab observations and our physical observations, we have our mathematical observations. And this may include things like hydraulic residence time, solid settling, but they will have kind of normal fluctuations. And that's why we kind of want to understand what is going on in our process and what our typical influent looks like. So examples of, when our influent rates change outside the normal range, here are some adjustments to troubleshoot and maintain influent quality. When the influent rate increases, there may be a higher organic load on the system. So to accommodate this, you should increase the dissolved oxygen in the aeration tank. Also, when the excessive flow rates increases, if the redundancy is available, it is beneficial to divert flow to spare clarifiers or aeration basins until flow decreases and the diverted flow can be treated. It's also important to increase the MLSS concentration or rate of return for return activated sludge, because this can benefit the activated sludge process when influent rates increase. Increasing the MLSS concentration in the aeration tank to maintain a sufficient population of microorganisms helps handle that increased organic load. And increasing the amount of RAS returned to the aeration basin extends the sludge age of the process as well, leading to more efficient treatment of those kind of shock loads. Longer sludge ages can also help improve the treatment efficiency and maintain stable operation when dealing with those higher organic loads. The older sludge age, they're more resilient to changes, and they've grown a little longer. They're usually more complex organisms as well. Now, if the influent rate decreases in the opposite fashion of increasing influent rates, there may be a lower organic load on the system. So when this happens, you should reduce the dissolved oxygen level in the aeration tank to avoid wasting energy. Lower oxygen levels will also help prevent excessive growth of microorganisms. And then again, in contrast to increased flow rates for decreased flow rates, you would want to decrease the MLSS or decrease RAS to the aeration basin and instead increase sludge wasting. This will maintain the proper biomass and decrease the sludge age of your system when you have decreased flow rates. Some more characteristics of our wastewater is going to be our TSS and our VSS. Just like BOD or COD in your influent, it can be difficult to account for an increase in TSS or VSS. But it is good to understand that typically you should have about 150 to 400 milligrams coming in, and the effluent should have about 1 to 20 milligrams leaving. And if that changes, there might be an issue with your system. But this is important to bring up because increased TSS and VSS can lead to issues such as bulking and foaming, reduced oxygen transfer, and a decrease in overall treatment efficiency. Adequate primary clarification is essential in controlling the total solids in your activated sludge process.
AJ, maybe I missed it, but can you tell us the definition of TSS and VSS?
I'm sorry, yeah, that's total suspended solids and volatile suspended solids. So this is gonna be like those particles that settle out in a primary clarifier for the TSS, and the VSS is the living portion of that. And also in our wastewater, it's important to understand, as we discuss these characteristics, some of the bacterial characteristics. So we have, like we said, our nitrifiers and our denitrifiers, which remove ammonia and then remove nitrates from the system. But there's also the heterotrophic bacteria, which primarily consume organic material. These are the organisms that will use our dissolved oxygen and our bound oxygen. And there's also the flock-forming bacteria. This is the filamentous bacteria that flocculate to form our biomasses. And these will extend like a skeleton through our biomass. So it's important to understand, what typically do these look like in our system, and if these start to change, what issues may arise from that. Now, after some of those other characteristics for our influent, there's also BOD and COD, our biological oxygen in demand, and our chemical oxygen demand. Like we said, this indicates how much oxygen the wastewater would consume if left untreated, and how the wastewater may impact the environment. So if you have an increase in BOD or COD, this typically requires more energy, microbial activity, and oxygen to treat. It's important to make adjustments to account for these types of increases and maintain proper sludge production to account for them. Again, though, it's hard to kind of make on-the-fly decisions about this due to analytical restraints. So it may require things like more increasing recirculation of your wastewater or adding more oxygen. Now, when you have untreated ammonia and TKN, this can result in eutrophication in receiving waters. This is what we were talking about with like algal blooms. Eutrophication is the excessive nutrients in those receiving waters that produce the excessive vegetative growth, which depletes the oxygen in wastewater and the eventual die-off of aerobic organisms. So the presence of these components in wastewater, and especially ammonia, is due to urine in the wastewater. Ammonia can be analyzed with laboratory analytics or a probe, and then simple lab results can be obtained in the field with grab samples and using a spectrometer in conjunction with the proper ammonia reagent. Now, when you have increased ammonia and TKN in your influent, it's important to increase the aeration and amount of DO in the process. That's typically the easiest way to account for elevated levels of ammonia. You can also do things like increase recirculation in the aeration basin, which helps remove excess ammonia in the wastewater. Both of these methods provide additional contact to dissolved oxygen that those nitrifying bacteria need to convert ammonia into nitrates. Nitrifying bacteria are very sensitive to environmental changes, and the nitrification process in general is sensitive to changes in pH. So if you notice temperature or pH changes in your influence, it is good to understand that could influence the nitrification of your system if it is designed to nitrify. Understanding that alkalinity can be used as a buffer to maintain the pH in your system is important. So in the event of an emergency, you can add alkaline buffers to the process. And it is also important to understand that nitrification is also known to consume alkalinity. and too much nitrification can lower the pH of your system. Now, in terms of other things that can be unusual in your influent, nitrate is a nitrogen compound found in wastewater and the byproduct of nitrification, like we've talked about. It is removed by denitrifying bacteria, which is our facultative bacteria, in the absence of oxygen. Large amounts of nitrate in water used as drinking water can result in methemoglobinemia, which is what we call our blue baby syndrome. And nitrogen removal typically is accomplished by having an anionic zone in your activated sludge process. Like we said, there's those denitrifying bacteria are facultatives and are facultative anaerobes, and in the process of, without the presence of dissolved oxygen, they use the oxygen bound to the nitrates. They're more readily consume the free oxygen, but if there's no free oxygen, they'll utilize the nitrates. Now, another thing to keep in mind, I see a question here, is 10 milligrams per liter the minimum contamination level? I think it depends on your permit, but for nitrates, yes. Typically ammonia, which you aim for 1. Now, something else that can come into your system, another influent characteristic to be aware of is your temperature, pH, and alkalinity. These are easily monitored with probes. And then, like we said a little while ago, it's important to monitor this because pH can affect your nitrification, denitrification, and overall microbial activity. Typical activated sludge plants run in the mesophilic range of 68 to 95 degrees Fahrenheit, and the typical pH range of 6.5 to 8.5 of what we typically see in like drinking water. Now, dissolved oxygen for influent is typically around 0.5 to 4.0. And like we said, electron provides that final electron acceptor. And if there's not enough dissolved oxygen in our system, it can cause issues with our BOD removal. DO issues that are typically associated with low DO, DO issues are typically associated with low DO, and this can be the result of a variety of causes. These issues can be observed observationally, like we said, with unusual bubbles. You know, if you start to see big giant bubbles, or a decrease in bubbles, if you start to see dead spots, and then also probes are used to monitor DO as well. Now, lower aeration rates or malfunctioning aeration equipment can result in insufficient oxygen transfer to the wastewater. It is important to first check blower equipment and valves when aeration levels are low. And then higher temperatures can also reduce the solubility of oxygen in water, make it harder for the water to hold dissolved oxygen. So as weather increases, it's important to kind of monitor your DO levels and see if you need to make any adjustments. Aeration equipment and diffusers can accumulate debris as well, such as solids and biofilms that can hinder the transfer of oxygen into the wastewater and result in low DO concentrations. That's typically associated with our diffusers or our aeration stones. And then also we've been discussing high organic loading, microbial overgrowth, and excessive solids that can consume excessive DO and can leave the activated sludge process with low levels of DO. Then finally, some issues that can cause the source of DO issues can be toxic shocks, such as the presence of a reducing agent chemical in the wastewater that consumes excessive DO. The next characteristic that we want to talk about and just introduce as part of some of our calculations is the mixed liquor suspended solids, and also our mixed liquor volatile suspended solids. MLSS, like we said, represents the concentration of organisms and other suspended solids such as inert particles and organic matter into the activated sludge process. Whereas the MLVSS represents the organic or living portion of the MLSS. Now, it's good to know what our typical MLSS is in our wastewater and in our influent. It's traditionally measured, it's typically about 1,000 to 4,000 milligrams per liter. MLSS is typically measured by filtering a sample and then drying at 105 degrees Celsius for 2 hours, and can also be measured with an electronic probe. MLVSS is measured by volatilizing the dried MLSS sample at 550 degrees Celsius for 1 hour. The MLVSS will be what is burned off when volatilized. That 550 degrees represents the temperature at which most living things will burn off those solids. We're about to talk about our F to M ratio as well, and MLVSS is considered the microbial portion of that activated sludge. So it is the M. And like we said, MLVSS is typically easier to monitor, or MLSS, I have too many acronyms going on right now, is typically easier to monitor because you can use a probe, while MLVSS typically makes up about 70 to 80% of MLSS. So you can kind of use an estimate, or if you have access to the lab information, you can utilize that as well. So that brings us to our F to M ratio. So our F to M ratio represents the amount of organic material entering a bioreactor compared to the mass of microorganisms available to consume the organic material. I guess I could have made a more distinct transition there. So we're kind of going from the characteristics of our influent and our activated sludge now to kind of our mathematical characteristics that we use some of those characteristics to calculate. And these are important to understand for both how our wastewater system is supposed to work and how it is operating at the time. So for that F portion, that is typically going to be our BOD concentration. And it's our BOT mixed along with the flow rate of influent into the activated sludge process. Then there's also the microorganism part, which is represented, like we said earlier, by our MLVSS. Now, the desired range for an F to M ratio is typically 0.2 to 0.6. And this is usually given as unitless, but it's really like our food to micro ratio per day. Now, monitoring the F to M ratio is essential to ensure the bioreactors are treating wastewater correctly and our permit requirements are being met. A low F to M ratio typically correlates with a high efficiency organic matter removal and longer sludge age. But low F to M ratios limit our microbial populations and decrease wastewater treatment. And they can also lead to, and when we have a decrease in our F to M ratio, this can be addressed by increasing our wasting, decreasing DO. Now, our high F to M ratio encourages the growth of specialized microbes that remove nutrients such as nitrogen and phosphorus. Our high F to M ratio leads to excessive sludge production, or it can lead to excessive sludge production, which can lead to treatment issues and contribute to unwanted solids. If you have a high F to M ratio, this can be addressed by decreasing sludge wasting, decreasing DO, and adding supplemental carbon sources. A lot of activated sludge processes, if they have a high F to M, if the F to M ratio is too high, this is in the wrong spot. It should be low. Low F to M, or activated sludge processes with a low F to M ratio will typically add a carbon source, such as things like dog food. So sorry about that. Under that bullet, that should be in the low F to M category above. And then one more thing to understand about the F to M ratio is that it's inversely correlated with the SRT and MCRT, which means that if the F to M ratio goes up, the SRT will go down and vice versa. To calculate our FTM ratio, this is typically done by dividing the mass of our food in the system by the mass of our microorganisms per day. This can be typically calculated by multiplying the influent flow times the BOD concentration over the volume of the aeration basin times the concentration of ml/VSS. So you can see here, here's our equation, and we added kind of what each of those variables represents. Now the units are typically going to be our food mass over our microbial mass, like we said, per day, and results in, because the masses cancel out, can be just represented as per day. As I left the 0.2 to 0.6 standard FTM ratio in there as well. Now, I'm going to show you a quick example of how we expand this equation out. So like we said, it's typically going to be, we don't typically know the loading, but we do know our flow and our concentrations. So by multiplying those out, we can get our correct units. Now, this can be done complicated by adding our conversion factors. So this would be done by like adding our 8.348 pounds. This, our per liter per milligram per millions of gallons. However, because you would do it to both sides, you typically just cancel it out to pounds of BOD for pounds of VSS. And the way this unit conversion works is our millions of gallons cancels out with our millions cancels out with our milligrams per liter. And this seems counterintuitive, but the reason is that milligrams per liter is equal to parts per million. So a part per million will cancel out the million in a million gallons per day or a million gallons. So that's where that goes. And like we said, because the unit's used on both sides, we can just have our pounds per BOD over our pounds of VSS per day. And if we actually remove that unit conversion, it just leaves us with our BOD over VSS per day. And that's why we have the ratio per day. So that just shows kind of that final inclusion of that equation. So I'm going to go over a quick example of this and show how we actually calculate this, though. It's a lot easier to see in function. So the FTM ratio, here's an example of that problem, how a typical problem might read on an operator certification exam. So this reads, the following data was collected for the performance of an activated sludge plant. What is the FTM ratio for this plant? Now, before I do this real quick, if anyone's worried about following along, we are going to send out the slides and a recording so you can work this out later for yourself. Or if you'd also like to attend one of our math trainings in the future, we have more opportunity to kind of sit down and kind of work these through with you guys. But so the first thing we need to do is identify what we need to solve for in the problem. What is the FTM ratio? And to do that, we need to also figure out what is our M that's going to come into place with that equation. So like we mentioned earlier, the ml/VSS is a component of our MLSS. So we actually multiplied that percent of solids times our MLSS concentration. So that gives us 80% times our 3100 MLSS, which results in 2,480 milligrams per liter. So we simply plug that into our F to M ratio equation, which is going to be our flow times BOD concentration over our volume of our aeration basin multiplied by the product of our volume of aeration basin, and the MLVSS concentration. So you can see we have 3.2 millions of gallons per day multiplied by 250 milligrams per liter. And then we have 0.9 million gallons multiplied by 2,480 milligrams per liter in our bottom half of our fraction. So the milligrams per liter cancels out on the top and the bottom. The millions of gallons cancels out on the top and the bottom. And that results in 0.36 pounds of BOD per pound of VSS per day. Then the other mathematical, or the next mathematical concept that we need to think about is our sludge age. So our sludge age indicates the amount of time that microorganisms or solids spend in the activated sludge process. So this ensures correct types of microbes are present and the wastewater treatment is optimized. So our sludge age is typically measured in days, and it's measured by the mass of solids in the aeration tank versus the mass of solids leaving the aeration tank. Or I'm sorry, in the tank versus what's actually entering the tank. No, that's not correct. I don't know why it says that. This should be leaving the tank. Now, operator certification tests and plant operations typically utilize more specific variants, such as our solids retention time and our minimum cell retention time. Sorry, guys, give me 1 second. I need to drink some water because I'm starting to cough after talking for 2 hours. Thank you all. Thanks for bearing with me. So that leads us to kind of discussing the solids retention time versus the mean cell residence time. So like we said, this represents the average time for the SRT. It represents the average time solids or cells are retained in the activated sludge process. And SRT is more commonly used than our MCRT. And it directly relates to the microbial activity, the nutrient removal, sludge production, effluent quality, and energy efficiency of our system. The SRT is determined by our design parameters, but the conventional plants typically have an SRT of 3.5 to 10 days. And this is typically controlled through our waste activated sludge rate and how much solids we are removing from our secondary clarifiers. By increasing our sludge wasting, we can reduce our SRT, and by decreasing the sludge wasting, that will increase our SRT. Now, for comparing the two, some certifications treat these two interchangeably, but some provide slightly different equations. From my experience, the Sac State course uses the SRT, and the ABC testing formula / conversion table, which a lot of our state tests use, treats them interchangeably. Now, the MCRT equations sometimes do not include the TSS in the secondary clarifier or only account for living solids. But this is our typical equation that we see. So it includes the solids in our aeration tank, the solids in our clarifier, and those are added together and then divided by the product of the solids that are wasted and the effluent solids. So that's why my wording was a little wrong on about 2 slides ago. It's because it's what's in the tank over what's leaving the tank. And if you use the metric system, this can also be represented by kilograms of what's in the tank over kilograms of what's leaving the tank. Now, activated sludge processes with only BOD have a removal, typically have an SRT range of 1 to 5 days. So that's in addition to what we kind of talked about earlier. Now, there are a variety of SRT equations adjustments that can be made. So, for example, or I'm just going to show the breakdown of how these units work out. So, like I said, or like we talked about earlier, milligrams per liter equals parts per million. So that will cancel all the millions in my multiplication segments of this equation. And then sometimes they do, like we said, they add the 8.34 pounds per gallon unit conversion to get to pounds. So that would cancel out my gallons. and just leave me with my pounds per day. But again, lots of times they just cancel out, and we just want to get to per day anyways. So if you, and then some of those adjustments that you made is if you do not utilize the TSS for the secondary clarifiers, or you don't know what it is, like if you don't measure that, a lot of times the equation might just include one MLSS and no solids for the secondary clarifier. And in that case, you actually apply the MLSS to both the volume of the aeration basin and the volume of the secondary clarifier. Now, if it's the kind of system, and this is more of an operational adjustment, if it's the kind of system that solids are immediately pumped from the secondary clarifier when you after the activated sludge, you would not account for that in your SRT equation. And then finally, some systems consider the solids in the effluent negligible, and in that case, you would really just multiply the volume or the solids that are in the aeration basin itself versus what is being wasted. So real quick, I can just show a quick example of that. So this is what that kind of a problem might look like in practice. You get an example of, SRT, given the following data for an activated sludge process, calculate the SRT. So you'd be getting all those variables that we talked about, the volumes for the aeration basin and the secondary clarifier, the flow of the influent and the wasting rate. You would have the MLSS concentration of the suspended solids, the solids concentration of the waste sludge, and the solids of what's in the effluent and what's in the suspended or of the secondary clarifier. Now to solve this, we always want to identify what we're looking for in the problem. So we simply calculate the SRT, and we find a problem that matches that. We're just going to use the standard one that I introduced earlier, and then we kind of just fill in those variables. And so that's what this might look like. Well, because we have it at our typical equations pound per day, we use the variables that we have, which is going to be our volumes, our flows, and our concentrations, and we can include that that unit conversion. But like I said, because it is because it's the same on the top and the bottom, mathematically it's just going to go away. So we can just go back to our equation because we know that our gallons, our millions, and our milligrams per liter are all going to cancel out and just result in days. So the actual equation, we'll leave it up there. And once we plug those variables in, it would look something like this. We have our 0.9 multiplied by our 3200 added to our 0.5 multiplied by our 540 concentration. That was our TSS of our secondary clarifiers. And then on the bottom, we have our influent multiplied. We use our influent rate as the same flow as the effluent because what's coming into the plant should be approximately what's leaving the plant. And we multiply that by our effluent concentration of solids. And then we add that product to the product of our waste flow times our wasting concentration. And that would result in 3.11 days. Then the next mathematical characteristic that we like to talk about is our SVI. Now with the SVI is we take a sample of our wastewater and we perform a settleometer test on it. We allow the settleometer test to go for 30 minutes and we measure the sludge volume after the 30 minutes. Typical volumes for this are 1,000 milliliters or 1 liter, but you can also use like 2 liters and stuff like that. Basically what tools are available to you. You just have to adjust your calculation. Now, SVI that is less than 80 typically indicates an old sludge age, while SVI that's greater than 150 typically indicates they might have filamentous issues and you may have improper bulking. Like we said, it's kind of controlled by adjusting your RAS and your WAS rates and optimizing what solids are in your system. So now more holistically, SVI is a parameter that's used to describe the settling characteristics. And like we said, you can use 1 to 2 liters, but really the goal is just to make sure that you understand where the sample is for 30 minutes. or after 30 minutes. And you want to go ahead and record the sample at 5 and 30 minutes. And this is the equation that we use to sample that. So you can see that the variables are going to be our volume at 30 minutes. And if you look at this, what that volume is the one on the right. So this would be about 250 milliliters. And so that's the volume that we use in that V30. So you have to have a clear wide mouth beaker to measure it. And you measure it at 30 minutes, and then when you start, which is typically 1 liter. You multiply that by a conversion factor, and then you divide that by your MLSS. So here's what that equation looks like in practice. So as you can see, we kind of just plug those numbers in. First, we find our 390 milliliters was our volume at 30 minutes. We have our 1 liter, which is what we used for the sample that we collected. And then we have our MLSS concentration that may be calculated in a lab or with a probe. And if you just solve the equation, you would get 150 milliliters per gram. Now, the last calculation that you'll typically use in activated sludge process is your sludge wasting. So sludge wasting is the process by which your excess biomass or MLSS is removed from your activated sludge process, and which we've been referring to as WAS. Now this can be variable, or this variable can be manipulated most readily to adjust our F to M ratio, our sludge age, our MCRT, and our SRT, because this is the most easy thing to control. Because we can't control how much water is coming in necessarily. We can't necessarily control the solids that's in that water. And we have to treat the wastewater to kind of account for like what solids are in the water afterwards. So the easiest way to manipulate those other calculations is to adjust our wasting rate. So like we said, this is the easiest thing to adjust for based on those.
AJ, we
yes.
have some questions in the chat right there. If you can see them, but
let's see.
Is there a worksheet or app that's used to get these results, or is it all handwritten solutions? Do all equations need to be this amount at all times? And basically what they're asking, what is the threshold amounts?
So, sir, okay, let's see. Program. A worksheet or app that's used to get these results. So this is what I would say. You can put this into a spreadsheet, Reddit, pretty easily. I'd be happy to help people with formulas for that. But the reason it's important to understand these calculations is because you're going to need them for your operator certifications. So it's good to know how to do both. Like you can set yourself up a worksheet pretty easily and plug these numbers in and kind of understand how your adjustments are changing and understand like your wasting target and stuff like that. But I would always recommend understanding how to plug these in and work and do it on a worksheet. So, or I'm sorry, do it handwritten, because you're going to need to do that to pass your operator certifications. And these numbers, so there's no threshold for like a wasting rate, but like we said with like SRT, like a traditional activated sludge plant would have about 5 to 15 days. Or if it's only used to treat BOD and it's more of a high rate, it would be like 1 to 5 days. So it's important to understand what specifics is required of your plant. And then if you are noticing issues with your activated sludge, it's not performing correctly, you can calculate these things to make adjustments as needed, say increase wasting, decrease wasting, that kind of thing. Sorry, I'm not trying to go too fast on here. But we do have not as much time as I'd like left.
We can't see your screen anymore. We see the tolls. Oh, there you go.
Yeah, sorry about that. So like we said, pumping is typically or increasing our sludge wasting rate or decreasing our sludge weight is typically the easiest way to adjust our other mathematical calculations and characteristics that we use for wastewater. So here's that equation that we use to do that. As you can see, it's like all the same variables, plus we have the MCRT is now on this side of the equation. And the MCRT you'd use is typically like your design MCRT to make sure that you are running properly, or if you're trying to decrease or increase your MCRT. That's the value that I plug in here. And now again, if you just remember, this equation adds volume and your volume of your aerator and your volume of your secondary clarifier, because we're going to be using your MLSS for both of them instead of just one. So an example of how you might see this on an operator exam would look something like this, where you'd have a problem that states data was collected for a conventional activated sludge plant that is experiencing decreased treatment efficiency, and the data is provided below. To decrease the sludge age of the microbial population in the plant manager would like to decrease the SRT to 6 days to more closely resemble the design specifications. What should the target sludge wasting rate be to accomplish this? So this gives us all our variables. We kind of carry that over to our equation, and we would plug those in. We have like what our SRT is that we would like to achieve. So we plug those in, and this would provide us about 70,500 gallons per day. Now, in practice, tripling the wasting rate could cause kind of some dramatic some drastic changes in your activated sludge, and you likely want to increase your wasting rate more slowly over a couple days. Just make sure not to get rid of your solids inventory. Now, we don't quite have enough time for this next part of the presentation. We have a scenario here where we can practice some of these equations. So I gave some characteristics and explained a problem, and we are going to ask for the F to M, SVI, and SRT. But we're going to go ahead and include these in the slides. And if you want to, work on these later, you're welcome to. And you have any questions, you're welcome to reach out to me, and I can help you if you have any issues trying to solve this. But with this is included, the three equations that it would that you would need, and what those results of those answers would be. So Matt, we're going to go and skip the next couple poll questions, because we're just going to go to our abnormal conditions and some safety factors. So with that, we're going to go and kind of talk real quick briefly about some common activated sludge process issues. These typically involve bulking sludge, foaming issues, settling problems, sludge overproduction, nutrient imbalance, organic overloading, and poor flocculation. So bulking sludge, the first one we're going to refer to is the abnormal growth of microorganisms, especially those filamentous bacteria in the activated sludge flock, causing the flock to become bulky, inefficient, and sorry, I had an alarm going off. Causing the flock to become bulky, inefficient, and difficult to settle. So bulking can result in poor wastewater treatment performance, such as reduced removal organic matter, nutrients, and suspended solids. There are a couple ways to control bulking. First, there's monitoring and process control. So the reason we need to know things like MLSS, MLVSS, or SRT, or F to M ratio, is we want to understand what it should be, and if it's out of those known parameters, we may want to decrease it. A lot of times things like a high F to M ratio may result in bulking sludge, because our sludge or our microorganisms that lead to kind of those issues have too much food, and they can grow out of control. The next one is foaming. So foaming is the formation of excessive foam on the surface of aeration tanks, secondary clarifiers, and other treatment processes. Similar to bulking, foaming is caused by excessive filamentous bacteria. The foaming is typically prevalent due to Nocardia and Microthrix, Parvicella. Those other filamentous bacteria that typically lead to foaming, but also surfactants, which reduce the surface tension and encourage foaming, can also be the cause. And just like in bulking, problems with foaming can be mitigated by ensuring proper MLSS concentrations, aeration, and nutrient balance in the activated sludge. Sometimes chemical agents can be used to remove the bacteria. A lot of times people use like powdered hypochlorite that's deployed in small controlled amounts. And then also anti-foaming agents can be used and mechanical scrapers. Now, to address surfactants, it's necessary to conduct community outreach campaigns to educate the public on surfactant issues and conduct pre-treatment monitoring to minimize the contribution of industrial discharge to the problem. And then finally, foaming is especially problematic during seasonal changes. So as you kind of go from that like winter to spring, summer to fall, you got to kind of monitor, kind of got to be extra aware that that could be an issue that may occur. And then also, as we discussed, sludge overproduction can be a serious issue in activated sludge and can lead to bulking as well. Increased sludge age, additional disposal costs, and reduced water quality due to issues with removing the solids from wastewater. Sludge overproduction can lead to bulking, but it should be noted it is not always the same thing, and it can just be an excessive biomass. Now, by optimizing your SRT and MLSS, this ensures that excessive amounts of sludge is not produced. And then also ensuring nutrients are balanced to limit the biomass growth is important as well. Then finally, optimizing your aeration, ensuring there is not excessive DO that promotes excessive biomass growth is important. Then there's also nutrient imbalance. This inactivated sludge refers to an unequal ratio of excess nutrients, particularly nitrogen and phosphorus, in the activated sludge process. As we've discussed, this can lead to bulking, sludge overproduction, and increased DO and energy overconsumption. To control your nutrient imbalances, operators should regularly monitor the influent wastewater characteristics to determine nutrient levels, particularly things for like nitrogen and phosphorus. This data is essential for identifying imbalances and adjusting treatment accordingly. So for plants that dose nutrients, the correct ratio of nitrogen to phosphorus is 5:1 to 10:1. Now, dosing of nutrient sources should be adjusted to achieve these nutrient ratios. In some cases, nutrients can be added as needed to balance the influent nutrient levels. For example, you can use chemicals like ammonia or urea to provide additional nitrogen, or you can add phosphoric acid or other phosphorus sources to supplement low phosphorus levels. Carbon sources should be optimized to create proper nutrient balance and ensure there is adequate supply of readily available carbon to act as food for the microorganisms. Carbon is essential for denitrification and can help balance the nitrogen to phosphorus ratio. And then finally, RAS can be increased to return biomass to the aeration tank to promote the removal of excess nutrients. The last one we're going to talk about is poor flocculation. This is just like kind of bulking and excessive sludge. This is often due to out of control filamentous bacteria or bacteria that's actually detrimental to those bacteria. Now, it's the reason that the reasons for poor flocculation may be improper mixing. So you want to make sure the mechanical mixing and aeration is adequate to ensure that flocculation is occurring. This is what proper mechanical mixing should look like. You can see that it's consistent, it's even throughout the basin, and there's no dead spots. Other variables to be aware of are variables in the collection system. Like we kind of talked about, this can be chemicals or your infiltration and your INI. And also seasonal changes can be a collection system issue that can lead to changes in your activated sludge process. Along with that, like we said, there's toxic loading that can occur due to chemicals from industries and homes. It's important to enact a pretreatment program and have proper education for your system and the community members to minimize the effects of toxic loading. Toxic loading also can be contributed by things like sewage haulers, and it's good to make sure that you understand with a pretreatment program what's actually being discharged from septic haulers into your collection system. We had one poll question, but we'll go ahead and skip it because we're just a minute after. And then real quick, I have a brief safety section. Some typical hazards to be aware of in your activated sludge is that aeration tanks, like we mentioned, they're more dangerous, or I don't know if I mentioned this, but these are more dangerous than unaerated tanks because aeration actually makes you less buoyant the more oxygen you have in a system. They've done like interesting, I don't know, like challenges to like Olympic swimmers to swim in like aeration tanks, clean ones. But they're putting that air in there, and because they're less buoyant, even Olympic swimmers have difficulty swimming in aeration tanks. So that coupled with like your rotating mixing equipment and the currents from those tanks make it a place that you do not want to fall in. If you do have to work near a tank, you want to make sure they have guardrails. That is an essential safety factor, and that there are life jackets available. You want to make sure that you have that the type of life flotation device you have are actually lifeguard approved with permanently attached lines to your surfaces. A good rule of thumb that is recommended is that there is 90 feet of line per life preserver. It's also important to remember that aeration tanks have other typical activated sludge issues, other typical wastewater treatment plant hazards, such as slippery surfaces, biochemical hazards. There's lots of moving equipment. There is going to be electrical hazards, and then also activated sludge processes can have excessive noise and confined space issues as well.